Thank you, Terry. Welcome, everybody. And so I'm going to talk about radioiodine therapy, but I'm going to do it perhaps in a slightly different way than you might have heard it before. Primarily focused on what we have learned from tumor genetics in terms of how that alters the biology of thyroid cancers, and in which way we can leverage that information to enhance responses to radioiodine therapy based on trying to manipulate the differentiation state of thyroid cells. So these are my natural relationships, and they are unrelated to what I'll talk about today. So, as you know, there's been now numerous studies exploring the genetic basis of cancer, primarily by using whole exome sequencing approaches. If you look at the overall mutation burden of different cancer types, and then you look at where papillary thyroid cancers fit within this, maybe I'll use the pointer. This is, papillary thyroid cancers are among the tumors with the lowest overall mutation burden. So this is, in general, a genetically relatively simple tumor type. If you now look at what the actual genetic lesions are in this disease, and I'm only talking here about papillary thyroid cancer, this is an oncoprint which essentially describes the different genetic changes in this tumor type. And just to orient you here, each one of these columns represents the sample of one patient, and each one of these rows is a particular genetic change associated with the disease. And the most common change is actually a mutation in BRAF, as many of you know. This is present in about 60% of all papillary thyroid cancers. Second most common are mutations in the 3 RAS genes, with NRAS mutations being the most frequent among these. And then there are a number of receptor tyrosine kinase fusions or other fusion events, including fusions of RAF kinase itself. And as you can tell, each of these essentially does not overlap with any other. So what this tells us is that a tumor that harbors a mutation in one of these particular genes gains no additional clonal advantage by selecting for a second mutation, which is an indicator that they probably have a pathway in common. And the pathway that they share is the activation of the MAP kinase pathway, which is common to all of these genetic lesions. The other factor to note here is that if you look at the overall copy number variance in this disease, that is, areas of chromosomes that have been gained or lost, most of these tumors are diploid. So essentially, it's a relatively simple tumor genetically, as I mentioned. Few genetic variants, primarily the driver mutations, few copy number changes. There are some exceptions I won't get into, but I think this is the fundamental message here. Now, because of this overall simplicity, the Cancer Genome Atlas study that essentially was a consortium of many different groups that was, assembled to study this disease, with each of the groups contributing different aspects of or different platforms to analyze these tumors, including the whole exome sequencing, as I mentioned, but also sequencing the messenger RNA. So this uses essentially RNA sequencing technology. And this, of course, was coupled to the information we knew about the histology and other particular variables, including a proteomic approach, which was a reverse protein phase array. And what was immediately obvious was that if you just looked at the gene expression clustering, which is what you're seeing over here, the tumors that harbored RAS mutations were quite distinct in their gene expression profile from those that harbored BRAF mutations. So the notion here was perhaps one could subclassify this disease in a way that's independent of knowing what the genetic lesion is by creating a score. Now, this is based on gene expression profiling of tumors that resembled more a BRAF tumor, whereas those that resembled more a RAS mutant tumor. And this is called the BRAF-RAS score. And essentially, it's a marker of the output of the MAP kinase pathway, with it being highest in BRAF mutant tumors and intermediate in RAS tumors. So if you now look at some of the other lesions and where they fall with this continuum, with this overall RAS-BRAF spectrum, what you can see is that there are certain BRAF mutations that are not the canonical V600E mutation, I'll get this back to this in a second, that fall within this, that behave more like RAS tumors. And now that the biochemistry of this pathway of these mutants has been studied, what we know is that they signal quite differently. So the actual specific mutation in BRAF matters, and I'll come back to that in a second. These actually signal more like RAS tumors because essentially they favor RAF dimerization. Similarly, as many of you may know, a fusion oncogene of Pax8 PPL gamma was reported many years ago as associated with follicular carcinomas, then found to be also present in follicular variant papillaries and in some follicular adenomas. So essentially they share histological similarity with RAS tumors, and indeed from a gene expression profile they also fit within this RAS-like picture. And then finally, and this is more, for people interested in this, this is a very interesting fusion found initially in thyroid adenomas, and that is also primarily associated with this follicular variant papillary thyroid cancer, which is primarily the histological type associated with RAS-like tumors.
Is that a gene?
Pardon me?
Is that the gene?
This is the gene. This is one of the components of the fusion. Yeah.
Because it's one of the type 2 diabetes susceptibility genes.
Interesting. Well, the fusion, I mean, this is the mechanism has been worked out by Yuri Nikiforov, who is a very talented pathologist who figured out that the abnormalities primarily through stimulation of IGF signaling, for a very interesting sort of story. So returning to this oncoprint, and again here you have the BRAF mutant tumors. If you now just specifically focus on the genes that are required for thyroid hormone biosynthesis, involving iodine metabolism. And if you look at in this heat map, whether they're expressed at high levels, shown in red, or to a lesser extent, shown in green, what you can see just by sort of eyeballing this is that tumors with BRAF mutations tend to have decreased expression of genes involved in iodine metabolism. Now, we knew this partially, but this gave us a much more sort of integrated perspective of this. By contrast, if you look at tumors that harbor either RAS mutations or RTK fusions, they have comparatively preserved expression of these genes. What we're not aware of, and the TCGA raised this issue, is that there is a cluster of BRAF tumors that paradoxically retain the thyroid differentiation function. And the mechanism of this is not well understood, but there are some early indications that some microRNAs appear to track with this. Some are actually going along and high in the context of this cluster or low in the context of this cluster. And there's several groups trying to understand whether these are actually going along for the ride, so essentially they're co-expressed with these genes, or whether they are actually determining some aspects of this different behavior of these BRAF tumors. Still unclear, but we should learn more in the future. So, putting all of this together, now connecting this a little bit more to the signaling, this clarity that you see in this disease in terms of what driver mutations do to gene expression, it actually fits within the overall continuum here of histology, biological behavior, and clinical behavior. So, for instance, BRAF mutant tumors primarily have a particular histological characteristic. They are either classical papillary thyroid cancers or tall cell variant papillary thyroid cancers. And if you now look at the way they signal, as I mentioned, BRAF V600E has this property that it actually signals as a monomer. Normally, just for a second, just turn to this middle panel, the way this pathway works is that upon activation of receptor tyrosine kinase, RAS is one of the proteins or one of the key intermediates that is activated primarily by GTP loading, and this in turn recruits RAF and allows RAF to dimerize. So the normal way that RAF signals is through dimerization. But BRAF V600E signals as a monomer. And because it does this, it is unresponsive to negative feedback loops that dampen the overall activity of the pathway. Because this negative feedback primarily it disrupts RAF dimerization. That's the way this pathway would normally attenuate the overall signaling output. So because of that, the overall output of the MAP kinase pathway is highest in the BRAF V600E mutant tumors. As I showed you just a minute ago, this high transcriptional output of MAP kinase is associated with a comparatively suppressed thyroid differentiation state. In the case of RTK fusions and RAS fusions, because these negative feedback loops are intact, or at least partially intact, this activity of the MAP kinase pathway is dampened, and their ability to impair thyroid differentiation is also attenuated. So, taking you a little bit from that human genomics to some functional experiments, primarily in mouse models, a number of years ago we just generated this mouse model to ask the question whether this overall phenomenon, first of all, of BRAF mutant state of these tumors, actually truly was causally related to impairing thyroid differentiation. And the second question was, is that a reversible process? And so, without getting into the details of the, the genomic tools we use to this, the notion here is that we generated mice where we had the ability, by treating them with doxycycline, which is artificially activating the expression of BRAF in the thyroid cells of these mice. So when you do this, 1 week after you switch BRAF on, you get a profound inhibition of the expression of this whole program of thyroid hormone biosynthesis. In fact, these animals became rapidly hypothyroid. If you then discontinue the doxycycline, you actually get restoration of their expression, and they become euthyroid again. So, but the fundamental reason we did this mouse model was to see whether we could achieve a similar result pharmacologically, not by switching BRAF, the gene itself, on and off, which clinically is not a feasible proposition, but instead, if we were to block the pathway, either with RAF kinase inhibitors or MEK inhibitors, whether we could achieve a qualitatively similar response. And the answer is that we did. And here we used iodine 124 PET imaging to look at uptake of iodine in the thyroid. 1 week after switching BRAF expression on, you can see that's decreased. When we wash out doxycycline, it's restored. And we get a similar effect when we treat with either a MEK inhibitor or a RAF kinase inhibitor. So that was the study that provided us the rationale to design this clinical trial that was led by Alan Ho, in which we asked the question of whether we could restore radioiodine responses in patients with RAI refractory metastatic thyroid cancer. So these were patients who had either no uptake on a post-therapy scan or a diagnostic scan, or had failed prior radioiodine therapies. So at baseline, these patients had an iodine 124 PET scan after stimulation with recombinant human TSH. And they then received the MEK inhibitor for 4 weeks. And whilst still on the drug, they had a second PET scan done, this is iodine PET, of course, and the endpoint was essentially to determine whether they had acquired sufficient lesional dose, whether we could predict that we could deliver a lesional dose of approximately 2,000 centigrade. That was the threshold. If we were able to meet that threshold, patients continued on the drug for another week and received a therapeutic dose of iodine 131. If, of course, they did not meet that threshold, the study was considered discontinued. So, since this was published a while back, and I think you may have seen it before, I'll summarize it briefly. It was a pilot study. 12 of the 20 patients actually had increased I-124 incorporation. I should say that the nice aspect of using iodine 124 is because it's a positron emitter, you can actually get a quantitative lesional dose, so you get an actual number, as opposed to using iodine 131, which gives you more of a qualitative change in uptake. And so this is a patient that basically had a RAS mutant metastatic thyroid cancer of the lung at baseline, and after selumetinib, while on selumetinib, another patient primarily with lytic lesions in bone before and after the drug. And of those 12 patients, 8 met that 2,000 centigrade lesion threshold and were treated with a therapeutic dose of radioactive iodine. And by RECIST, measuring the lesions 6 months later, all of them achieved some size reduction, and of these, 5 met the criteria for a partial response, the oncological criteria of confirmed RECIST response. So when we then looked at their genotype, what we found was that those patients that had RAS mutant disease, of which we had 5, all of these met that lesional threshold, and actually all of them I think 4 of these 5 responded very nicely to radioactive iodine. Whereas those patients with BRAF mutant disease, 4 of the 9 only actually increased their radioactive iodine uptake, and only 1 of 9 met the threshold for treatment. So we did not do very well with BRAF mutant thyroid cancer. So the question was, what was the explanation for this? So if we now look at just another series of studies focused specifically on the response of BRAF mutant cancers of other lineages, other tumor types, to RAF kinase inhibition. Let's look first at the responses of patients with thyroid cancer. This is a study by Marsha Bros, unrelated to radioactive iodine. So this is patients with metastatic BRAF mutant thyroid cancer. They are given a RAF kinase inhibitor, and then you look at their responses. And so you can see this is their best overall response, and overall the objective response rate was about 38%. If you compare this to the responses in patients with metastatic BRAF mutant melanoma, you can see that the responses are much better. Here it's 53%, and I should say many of these patients, I would say the overwhelming majority of these patients, were extensively pretreated and had failed a number of other prior therapies, which was not the case in thyroid cancers that were more virgin to systemic therapies prior to that. So, lineage differences in response. So, we were interested in trying to understand what might explain this, and I'm going to explain this relatively quickly, but it's a study primarily in cell lines, mostly poorly differentiated anaplastic cell lines. And if you look here at the left panel, these are a number of different BRAF mutant cell lines from either melanoma, from thyroid, or from colorectal, which, like thyroid, do not respond very well to RAF kinase inhibitors. And as you can see, whereas melanoma cell lines have a very nice inhibition by venurafenib with an IC50 of about 100 nanomolar, most thyroid cancer cell lines do not respond at all. Most colorectal cell lines actually don't do very well. And of course, if the tumor does not have a BRAF mutation, they do not respond. If you now look at this middle panel, what you're looking here is the phosphodynamics, or the response of the pathway, the MAP kinase pathway to inhibition over time after you expose them to the RAF kinase inhibitor. So these are melanoma cell lines. You treat these cell lines with the drug, and this is phosphorylation of the downstream substrate in MAP kinase, ERK, phospho-ERK. And you can see that when you treat them, they have a sustained inhibition of the pathway. By contrast, if you look at thyroid cancer cell lines, they have transient inhibition of the pathway, about 1 hour. By 6 hours, they rebound, so they come right back. So they essentially have an adaptation to the effect of blocking this pathway with venurafenib. So we asked the question why, and without showing you all of the data, essentially we did a screen to find out. Our hypothesis was that they were de-repressing inputs upstream of RAF. And this was proved to be the case in that we had a markedly increased phosphorylation of this member of the EGF receptor family, HER3, which we found through a phospho receptor tyrosine kinase array. And indeed, if you look at a time course after exposure to venurafenib, there was very robust activation of this receptor, and this was associated, as I showed before, with the activation of ERK and AKT signaling. Now, this was not just in one cell line, this was a lineage phenomenon. So if you now look across multiple thyroid cancer cell lines, you can see this activation of HER3 in response to venurafenib was common to the great majority of them, but was not present in melanoma, colorectal cell lines. So they respond differentially to inhibition of the pathway. So the issue is lineage actually matters. So you would predict then, if you were to block this, you would achieve an improved response. And HER3 essentially it has no intrinsic kinase activity. It binds to other members of this family, primarily HER2, which was the main partner that we found to be involved here. And so we used an inhibitor of HER2 kinase. It actually also inhibits EGF receptor kinase. So if you look at just the exposure to the RAF kinase inhibitor, you get this, again, robust activation of HER3 and a rebound activation of phospho-ERK. But if you combine this with an inhibitor of HER2 signaling, you actually block the rebound and you also block the adaptive response. And the similar effects that I won't go into in detail in the genetically engineered mouse model that we had studied. So this is essentially what we found in this paper. It was published also a few years ago, in which when you think of the tumor, untreated, you have this robust activation of MAP kinase signaling. This in turn tends to repress inputs upstream of RAF. It's a normal feedback response to try and dampen the overall output when you have a signal that drives the pathway. In the case of HER3, it does it by suppressing the actual expression of this receptor through interaction with some inhibitory transcription factors. When you come in with either a RAF or a MEK inhibitor and you block this pathway, this upstream signal gets de-repressed, in part because transcription of HER3 is increased, but these cells are unfortunate enough that they constitutively make the ligand for this HER2-HER3 dimer, which is neuregulin 1. So in response to this, you get reactivation of the pathway and adaptive resistance. So we wanted to now test this in a clinical trial, and this was led by Eric Sherman and reported last year, in which we combined these two drugs now in patients. In this case, dabrafenib, another RAF kinase inhibitor, and lapatinib, which is a HER2-HER3 inhibitor. And this was a phase 1 trial because this combination had not been studied previously, so it was essentially had safety as its primary endpoint. So in which we used a common dose of dabrafenib with increasing doses of lapatinib, as shown here. Essentially, surprisingly well tolerated. And these were the overall responses. So what you're looking at here is all patients, and then Eric excluded the patient with anaplastic thyroid cancer because, in a way, we did not expect much response there. You just see the responses here. These are the 3 doses, the number of patients studied at each dose level, and this is the overall median progression-free survival, which was 15 months, and the response rate, which is 60%. Compare that to the 38% response rate that Marsha Brose published with a RAF kinase inhibitor alone. And if you exclude the anaplastic thyroid cancer, the response rate was 70%. So clearly, a significant signal for improvement. See, and this is just essentially the waterfall plot. We don't need to go over it. But these are actually profound responses. This is a significant change. And some individual examples here of shrinkage of metastatic lesions in the lung and in brain. Okay, so with this, I think what I've tried to show is that thyroid cancers, this particular lineage, tends to have a very robust adaptation to monotherapies that block MAP kinase signaling. So the question is, what about responses to reactive iodine? That initial study that I mentioned of selumetinib had shown very weak responses in the BRAF cohort. So we were interested in trying to see if we were able to achieve a more comprehensive blockade of the pathway, whether we might get improved responses. So selumetinib is actually an inhibitor of MEK, as I mentioned, and it's an allosteric inhibitor. So essentially, most kinase inhibitors that you see that are sort of now approved for many different tumor types are primarily ATP competitive inhibitors. These kinases have to bind ATP in order to transphosphorylate their substrates. And their selectivity is essentially designed by trying to look at variations within that ATP binding pocket that give a drug a particular selectivity towards one kinase versus others. This drug is different in that it does not bind to the ATP pocket, it binds to a different surface of the molecule. And a company in Japan, Chugai, developed another allosteric inhibitor that binds to the same overall site in MEK, but has this unique property that it sequesters and also indirectly inhibits RAF kinase activity. So in practice, it functions as an inhibitor both of RAF and MEK. And this is essentially the data proving this in vitro kinase assays showing inhibition and equimolar levels of both MEK and RAF kinases. So we have a model that we developed of papillary thyroid cancer driven by BRAF V600E. It's a genetically accurate model. It's a knocking mutation, uniquely in the thyroid. It has high penetrance of papillary thyroid cancer. So by 3 weeks, essentially all these mice develop papillary thyroid cancer. So what we did here is we wanted to see, first of all, the phosphodynamics of this pathway. So when we treat with selumetinib, just as we saw in the cell lines, I'm sorry, these are Western blots. Each of these lanes is the thyroid of one mice, either treated with no treatment at all or vehicle, and then at various times after exposure to the drug. This is selumetinib. This is the Chugai compound. And as you can see, when you treat with selumetinib, you get a transient inhibition of ERK signaling followed by a nice rebound. Well, nice for the gels, not for the treatment response. But if you look at the combined RAF-MEK inhibitor, you get a much more sustained inhibition of the pathway. If you now look at the treatment for 1 week, either with vehicle or untreated here, with a MEK inhibitor or with a Chugai compound, you can see that you get more profound inhibition of ERK and a greater restoration of expression of the sodium iodide synthase. Now, a better way of quantitating output of pathways is actually looking at their transcriptional readouts. So, what has been shown is that if you want to kind of have a sense of the activity of the pathway, these phosphorylation plots give you a notion of how the pathway is switched on and off, but are not quantitative. But you can get integrated information by looking at a number of transcripts that are regulated, in this case, by MAP kinase. And these two in particular happen to be very faithful reflections of the overall output of MAP kinase signaling. So if you look at the differences, go back here. Here, what you're looking at is the percent of vehicle. So essentially, this is the BRAF mutant papillary thyroid cancer treated with vehicle. We call this 100% expression of these two genes. And looking at various doses of the Chugai compound and then the maximum dose of selumetinib. And when you treat with selumetinib, you get roughly a 70% inhibition of the pathway. When you treat with a dual inhibitor, a RAF and MEK, you get an additional decrement of another 15%, so 85% inhibition. That relatively small difference results in major changes in the re-expression of differentiation factors, such that with selumetinib, you can only restore the sodium iodide synthase to a very modest extent, the same for thyroglobulin, whereas when you treat with a dual inhibitor, you actually get a much better response. And we followed this by essentially treating these animals, first doing I-124 PET scans, the same that we did with the clinical trial, and with selumetinib, you get a very modest increase in iodine incorporation in the lesional dose that you can achieve. And then if you look at the treatment after 6 months, so remember these mice receive drug only for a short period of time, then they get the treatment with radioactive iodine, then there are no treatment whatsoever, so you're just monitoring response to the radioactive iodine treatment. You only have a vestigial remnant of the tumor in the Chugai compound treated animal, where with selumetinib there's still persistent disease, somewhat better as you see here in the ultrasounds over time, but you really don't get anything like this response. So the message here is essentially that in order to maximize the ability for us to redifferentiate these tumors, you need to inhibit MAP kinase pathway profoundly. So the initial study, we had just used a MEK inhibitor. This is a study designed very similar to the one before, but the only difference is that here we're actually trying to determine in patient biopsies what are essentially the changes that we see over time. So these patients have RAI refractory metastatic disease, treated now with the RAF kinase inhibitor, vemurafenib, and in these patients we did biopsies at baseline and while on compound. First of all, and so the way we decided to monitor this is that is to develop a platform which is called NanoString platform. So this is essentially an ability to measure multiple messenger RNA, multiple transcripts that you can do in paraffin-embedded tissue. And it's using an approach that essentially has two different probes that bind to the primary mRNA target. So the first is a capture probe. It binds to one site in the molecule. The second is the reporter probe that binds to the same messenger RNA at a different site, relatively contiguous to the other, but has this barcode here. And this barcode allows you to actually measure 200 plus transcripts. And we designed it in such a way that we could detect what we call the enhanced thyroid differentiation score. So there were 16 genes that were used by TCGA to define this thyroid hormone biosynthesis pathway. To this, we added genes that correlated either positively or negatively, again from the TCGA data, and we essentially are trying to monitor this thyroid differentiation state of the cancer cell. And in addition to that, we use this BRAF-RAS classifier that I described earlier, the genes that define those particular groups, as well as those a series of transcripts that determine the ERK output, and then various controls. So these essentially were the results. Where we had only 3 paired biopsies. This was a pilot study again, and what you're looking at here is this enhanced thyroid differentiation score. So this patient here had the best response. You can see here sort of a nodal recurrence in the neck, which is RAI non-avid at baseline, and then has a very robust incorporation of iodine in response to the drug. This patient with metastatic disease of the lung had an intermediate response, and this one had a very attenuated response here. And we actually saw a very corresponding change in this signature of thyroid differentiation. At baseline, the best responder, in fact, had more preserved thyroid differentiation state at baseline and had a more robust response. Intermediate for this patient and lowest here. Now, the idea, this in a way is not entirely surprising. You would expect more iodine incorporation, better expression of these genes. But the idea here is, could the baseline information eventually provide us the information that will tell us which patients are more or less likely to respond to these therapeutic strategies? And that's a question we don't have an answer for, but that we are trying to pursue. So comparing then these changes, this is what I showed you before with selumetinib, 4 of 9 responders, 1 of 9 that increased incorporation, much better in the case of 4 of 9 that increased incorporation, 1 of 9 that responded. It's, clearly better with vemurafenib, but of course very small numbers. And just, one of the issues that we contend with in these clinical studies is that it's very difficult to interest companies in doing larger studies. We are, continually trying to get sort of interest in trying to do a more definitive study that will allow registration of some of these compounds for this use for metastatic disease. There is an ongoing phase 3 trial for selumetinib in the adjuvant setting. So these are patients who went to thyroid surgery, were considered to be high risk for recurrence based on their nodal status or the primary tumor size, and they received either placebo or selumetinib prior to getting conventional radioiodine. And in those particular, so the response that we should find out what the result of that trial are sometime later this year, we hope. And again, Alan Ho was the principal investigator in that study. So what I've said so far is this inverse relationship between the activity and the transcriptional output of MAP kinase, the expression of thyroid differentiation markers, and this translates into distinct RAI activity. So the dirty secret here is that we have a problem with this approach. If you just look at the, what we know in terms of the overall thyroid hormone biosynthetic pathway, as you know, in the basolateral surfaces where you actually have the sodium iodide symporter, incorporates iodine. This needs to be organified to be incorporated into thyroglobulin, into tyrosine residues of thyroglobulin. For this to happen, the iodine needs to traverse the apical surface, get oxidized through peroxide that is generated through these dual oxidase enzymes. And then thyroid peroxidase catalyzes the incorporation of this iodine into tyrosine residues of thyroglobulin. These then get re-imported into the cell and eventually secreted into the circulation. Now, what I showed you before was that when we treated these mice with BRAF mutant thyroid cancer, you might notice I treat mice and humans the same. So that essentially they're about the same for me. So when we when you look at the iodine uptake, as I showed you before, we show this beautiful, nice increase of iodine incorporation when you had a more potent inhibition of the pathway. But if you look at the iodine retention time, there's no change. And in order to get your maximal effective rate of iodine, you don't want it to go in and out. You want it to be incorporated into protein. And the determinants of that, as I mentioned, are these enzymes. And these two particular enzymes, DUOX1, DUOX2, are actually not restored by these drugs. So when you inhibit in a BRAF-mutant tumor the activity of the MAP kinase pathway, one of the adaptive changes is that besides reactivating MAP kinase signaling, you also activate the PI3 kinase pathway because it's driven by receptor tyrosine kinases. And both those pathways, among others, are also switched on. And an example of that is shown here. This is a vehicle-treated mouse with papillary thyroid cancer. And this is a RAF and MEK inhibitor. It's another study where we combine these two drugs. And if you just look at the expression of a readout of the PI3 kinase pathway, you can see that that is very significantly increased across the board. And that's shown quantitatively below. So we have asked the question about whether, if we actually were to inhibit both of these pathways, whether we would have a different effect. Same design, mice at baseline, I-124 PET imaging, and then while on this combination.
Just for of all the PI3 kinase inhibitors, pick one I never heard of, so that don't say something about me. I'm wondering why you think that.
This is a pan-PI3 kinase inhibitor, so it inhibits all of the different types. So we wanted to ask the broad question first, and then think more selectively later. But it's a good question because these two drugs have been tried in patients. They're very toxic. When you combine inhibitors of MAP kinase with a pan-PI3 kinase inhibitor, drug companies want to know nothing more about that because the responses were, the toxicity was too high. So if we now just look at the disappearance time of I-124 in mice treated with just the RAF kinase inhibitor, it just essentially goes down very quickly. This is in a wild-type mouse, so there's no thyroid cancer here, obviously untreated, and this is the retention time. And with the combination, we actually have had a markedly prolonged retention time. This is an example, and I think it's pretty representative. At baseline, this is just the PI3 kinase inhibitor by itself, it does very little. The combination here, 66% of the isotope was retained in 72 hours, so it was really striking. So the question is, well, had we really changed the expression of these two genes? And the answer is that you did. So again, with the sodium iodide importer, you beautifully restore it with the Chugai compound. The PI3 kinase does nothing, maybe a little bit better with a combination. This is thyroglobulin, this is thiroperoxidase. This is the dual oxidases that are not restored by MAP kinase inhibition, but when you use the combination, you get very nice restoration. Let me just skip that. So if that were the case, you would expect more iodine to be incorporated into thyroglobulin. And so what we did is we harvested these thyroids, and after giving them a dose of iodine 131, and did an autoradiogram to try and see whether the iodine had incorporated into protein, and specifically into thyroglobulin. And the answer is that they did. This is the Chugai compound alone. These are the PI3 kinase inhibitors alone. Here, yes, either selective alpha, beta, and pan. And by themselves, they do nothing. But if you look at the combination, it's significantly increased, and that's quantitatively shown here. So where we are with this, as I mentioned, pan-PI3 kinase inhibitors are not feasible in combination. But so now we're looking at the selective inhibitors where any of these, which are better tolerated, can be combined to try and achieve a better response. So in the final 10 minutes or so, what I'd like to do is to maybe sort of get a reality check here, because it's correct that papillary thyroid cancer are genetically simple tumors, and they retain this dependency on the initial driver to a remarkable extent. There was a study recently published showing that anaplastic thyroid cancers with BRAF mutations, when you use the combination of a RAF and a MEK inhibitor, they had remarkably good responses, transient. So you get like a cytoreduction effect. Of course, the disease comes back. So the issue is, when you have more complex tumors, when you have tumors that just don't harbor the BRAF mutation itself, but harbor other gene changes, will this work? Or actually, maybe, are we selecting for a more virulent clone that might develop later over time? So, shown in a model format, if you treat with a RAF or MEK inhibitor or a combination, then you give radioactive iodine, you get a nice response. You eliminate most of these green cells that trap iodine very well, but you're left with these other cells that can eventually evolve and grow. And so the question is, what might be those secondary genetic changes? And we decided to focus on a series of these in particular. So this is a recent study that was published a year or so ago, in where we did studies of the genomic landscape of polydifferentiated thyroid cancers and anaplastic thyroid cancers. So these tumors, of course, as you might expect, have a much higher mutation burden. They're genetically much more complex. I won't go through every aspect of this, but the drivers are actually quite common to the papillaries, and that BRAF is the most common RAS, enriched compared to papillary, but of intermediate frequency. Fusion oncogenes, that is RTK fusions, are seen in polydifferentiated thyroid cancer, but are very infrequent in anaplastic cancers. What they do have is a very high frequency of mutations of the telomerase promoter, and that probably is involved in immortalization. And as we and others have reported previously, P53 mutations are a hallmark of anaplastic thyroid cancers, but they're only present at intermediate frequency in polydifferentiated tumors. What we decided to focus on, for a number of reasons, were genes that are involved in chromatin regulation, and specifically this particular complex, which is called SWI/SNF. I'll explain what it is in a second, where mutations happen with intermediate frequency in polydifferentiated thyroid cancer, but very common in advanced disease. So the reason we were interested in this is that in parallel to this study, we had done a forward genetic screen in mice. So essentially we used an approach where we used an immunogenesis approach to try and find genes that cooperated with RAS and transformation. And these SWI/SNF genes proved to be very significant hits in this particular study. So based on that evidence, we decided to focus on this particular complex. And here you see examples of the mutations that we found in anaplastic thyroid cancer, polydifferentiated thyroid cancer. These are different members of this complex, and these are mostly in black truncation mutations. So they're clearly loss of function mutations. And this is their overall frequency. It's about 36% in anaplastic. It's lower in polydifferentiated thyroid cancers. So the nomenclature of these complexes is complicated because two groups call them differently, but bottom line is there are two main complexes. One is called BAF, the other is called PBAF. And essentially they have ATPase activity, and what they do is the following. Sorry, before I do that, I'm a little ahead of myself. This again is the overall frequency of these SWI/SNF mutations in polydifferentiated anaplastic thyroid cancers. And just to prove the evidence that these are actually involved in tumor microevolution, if you look at their clonality, what you can see is that in papillary thyroid cancers, you already begin to see a few of these tumors that have these mutations, but they're subclonal. They're only a small component of the tumor. But as you look at polydifferentiated or anaplastic thyroid cancers, then they become clonal. It's the same thing for SWI/SNF, for P53, for TERT. So all of these lesions involved in advanced disease are present, if you sequence deep enough, in a subset of well-differentiated thyroid cancers. And they would have that propensity to emerge as resistant tumors or more advanced tumors later on, which, obviously raises the question is, should we be doing some deep sequencing in some of these patients to predict for these things? But that's a separate discussion. The other aspect of this is that if you look at these different components of this SWI/SNF complex, and in different diseases, they've been found to be mutated and play an important role, but they tend to be a dominant member of each of the complex that's primarily defective in these diseases like renal carcinoma, endometrial carcinoma, rhabdoid cancers, and others. But in thyroid cancer, you essentially see mutations in multiple of these components with roughly equivalent frequency. So we decided to take a systematic approach to look at this by essentially disrupting one member of each of these complexes, which is Iod1A and Iod2 for BAF, PBAF, and this one that disrupts both complexes. complexes. So it disables this machinery altogether. And just to cut the long story short, they're associated with very rapid development of advanced disease. They develop poorly differentiated anaplastic thyroid cancer. Here's the timeline. It's actually very, very clear. And they're associated with significant mortality. So what do these complexes do? So these essentially are multi-subunit complexes, as I mentioned, that are involved that use ATP hydrolysis to mobilize nucleosomes. So as many of you know, nucleosomes are areas of the genome that are compacted. So the DNA is essentially wound around these histone proteins. And these areas usually are not accessible to transcription factors. So the areas of the a genome that are available for transcription are those intervening sites that are not actually compacted in these nucleosomes. So what SWI/SNF essentially does is that when it enters into these areas, is able to essentially disrupt this nucleosome organization, open up chromatin, and allow transcription factors to access. Tends to be the simplistic model, which is now, it's still correct, but essentially not universally so, is that there is essentially two different complexes that compete with each other. What maintains these nucleosomes in a compact state, and therefore in a more stem cell-like state, are members of this complex called polyclonal repressor complex. And when SWI/SNF comes in, that complex goes out and vice versa. And I've got to say that there are multiple exceptions to this. So the first question that we asked when we had these mice was, well, what does the expression of these genes, or the loss of their expression, due to the differentiation state of these tumors? So here on the left, what you're looking at is, this is the BRAF mutant tumor, just BRAF mutation. And here's the expression of the thyroid-specific genes in a wild-type mouse and in a BRAF mutant mouse. And as I showed you before, these are decreased. But if you now take the BRAF mutant mouse as a baseline, and you, let me correct myself. If you now look at the effects of each of these three disruptions, you see these two are associated with a decreased expression of these genes. And if you look at them in combination with BRAF, their inhibition is much more profound. And I just want to show you some illustration of where we're headed with this and why we're interested in this problem. So again, here you have areas of closed chromatin, not transcription accessible. And you can actually monitor and find these areas of open chromatin using this technique called ATAC-seq, which is essentially a way in which you can get these transposomes that can get into the areas that are not compacted, excise them into multiple tiny fragments, and because they actually have two adapters, you can sequence these by deep sequencing. So you can actually find peaks of sequencing wherever there are no nucleosomes. So this here is a key transcription factor that's involved in thyroid differentiation, which is called PAX8. And here you have the gene. And these are two peaks, presumably associated with enhancers for PAX8. And then you can see very nicely present in a wild-type thyroid, but totally gone when you actually disrupt these three different genes. This is NKX2.1, which is the second transcription factor, also involved in thyroid specification. Again, you see a decrease in these overall peaks, here primarily within the gene body. And this is the sodium iodide symport. So at baseline, you can see these nice peaks within the gene itself, within the promoter. And there's also a site actually reported by Greg Brent 10 years ago as a sodium iodide symport enhancer in rat PCCF3 cells, where we also see a beautiful peak. And these are lost when you lose the expression of these genes. Anyway, so final data slide. So what does this do to our ability to redifferentiate thyroid cancer cells in these mouse models? So again, we're using the RAF MEK inhibitor. And here you have a BRAF mutant mouse at baseline. These are the expression of thyroid-specific genes. This is with ARID1A knockout or with SMARCB1 knockout. At baseline, you can already see that this particular disruption that shuts off all SWI/SNF complexes have an even lower expression of these genes at baseline. If you now treat with the RAF-MEK inhibitor, nicely restore their expression of these genes in the BRAF mice, you have an intermediate response, but unfortunately not for the sodium iodide symporter in the ARID1A mouse. And if you have no SWI/SNF, if you disrupt these chemicals altogether, these are irreversibly dedifferentiated. So the idea here is there's an important interaction to be studied between what can be manipulated by altering the signaling of the driver with what happens in terms of epigenetic states that essentially could disrupt the access of transcription factors directly into these sites. And these are areas that we're interested in pursuing because the story is not totally hopeless in that when you have these interactions, other complexes responsive to enzymatic inhibitors can access and repress these sites, and these could be amenable to pharmacotherapies as well. So I think I'll stop at this point. I tried to have acknowledgements of the people who did each aspect of the work within the slides, but most of I'd like to highlight in particular my colleague Alan Ho, who is a MD PhD medical oncologist who leads many of the clinical trials that I showed you today. That's really his work primarily, and as well as Eric Sherman, who also is heavily involved in this. In my lab, Inigo Lam has been the primary person involved with the genomic studies. I showed you data from a former lab member, James Nagaraja, who's a nuclear medicine physician from Germany who did many of the I-124 PET imaging studies in response to different drugs. And the evolving story on the epigenetics of thyroid cancers done by Javi Leandro Garcia and Hesh Saxena. And I think we have a few minutes for questions. Thanks.